So Alison Robertson from Dauphin Island Sea Lab is going to be talking to us next about things like forecasting and tools like that, where we are, where the state of the science is at, what we can and what we cannot do. So,
yeah, awesome. Thanks. Hi, a lot of you locally will know me. I've been working here on the Gulf Coast on HABs since 2009. So I am familiar with the area, I'm familiar with the problems, I'm familiar with the oceanography, so we can talk a bit about those issues and problems and any questions that you might have. But I've been working on HABs for actually 20 years now, which is a bit scary to say and makes me feel like the grey hair is warranted. But so I've worked on HABs all over the world in different countries. Everybody is dealing with this in a similar way. So the best thing we can do actually is collaborate with everyone around the world, which is why I think the efforts that have been made in the past are actually really, really important, really informative. But to get a really good predictive tool and a really good model, we have to work on local conditions. So a lot of the forecasting tools that have been developed are fantastic. A lot of effort and science has gone into that. But of course, things are a bit different when you start looking at a local scale. We have a huge freshwater import here from Mobile Bay that changes the way things work, that changes the way things move, that changes the duration and propagation of these HABs, and it changes the nutrient dynamics in the system, which is important in trying to understand how these things are going to work. So I'll go through some of those things, but again, I kept this fairly simple, so please ask more questions if you have them. I'm happy to answer any of them. Something I have not included, but I'm happy to discuss, is control and mitigation strategies, so solutions that we could potentially apply. That's not part of this talk, but I was going to leave that open for a discussion. So I don't know why there's a weird little patch on the screen, but that's okay. We'll just roll with that, okay? That just makes it like more enticing maybe to look up there. I just want to always remind people that phytoplankton are actually critical to life on the planet, producing more than 50% of the world's oxygen. So these are actually the good guys. It's just very, very few that are harmful. So we do need phytoplankton in our ecosystems to have a stable and viable ecosystem. Oysters feed on phytoplankton, so it's a necessary food source. All of those things are important to remember when we start thinking about control and mitigation and modeling of these things, because there are a lot of really, really good phytoplankton that are in the system, and the same satellite imagery are going to pick up those particular species as well as the harmful ones, but not necessarily discriminate between them. So sometimes we can be chasing the good guys, thinking we're trying to protect ourselves from the bad guys. So that's just an important point to make. Locally, and those of you that have seen some of the work that we do in my lab before will have seen something like this, but I'm trying to, okay, pointer is here. At the top here, just show you some of the local HAB species that are relevant. So we have, we've been talking about red tide, and of course I'm going to focus on that, but there are others in our system that you need to be aware of, we need to think about, and the way that they respond to different environmental drivers or different environmental factors and conditions is all quite different. So they all have different lifestyles, I guess you could say. So we have Karenia brevis at the top up here, and we know a lot about it, causes red tide. Usually, in conjunction with Karenia brevis, we also have Karenia miki-moitoi. So it's another species of Karenia that also can produce toxins. So it's important to know that there's not just Karenia brevis here. We do have others that bloom at the same time usually. So it's mixed. The one that I do want to note just quickly, and the pointer isn't really working, so we'll just get action over here. So the Caladidian little cell that's in the middle is a little bit hard to identify from this image because you'd have to rotate the cell a little bit, just plop it over in a different way. It's actually the cause of most of the fish kills that happen in Mobile Bay. So every week I will get a call about fish kills. Sometimes that's related to hypoxia, so low oxygen in the water, and sometimes it's because of this particular species. It is not a problem for human health, but it is an ichthyotoxin, so that means it can kill fish. It does that by clogging up their gills. So that can happen. So when fish kills are seen, you don't have to assume a big bloom of Corenia has come in and destroying everything. Although that's possible, because Corenia can cause fish kills as well, but it does so through the dissolved water, the dissolved toxin content that's in the water that gets into the fish. So then we have Pseudonisha, and have a very great colleague in the room here that studies silica, so he will appreciate this one. But Pseudonisha is a diatom. It is actually stimulated much more rapidly in bloom conditions. Nutrient levels required for these particular organisms to bloom are very different from Corenia. And then down in the bottom corner here, this big thing with a nice tail around it. Diaphysis cordata is the cause of diuretic shellfish poisoning. And it is highly relevant here. We see this all the time, but it's quite patchy. And this organism doesn't usually bloom on the surface. It blooms sort of in the middle of the water column. So it can, again, be difficult to get any kind of satellite imagery from, and a bit difficult to predict. The levels for regulation are also much lower for this particular genus or this group of phytoplankton. So very low levels can produce harmful toxins. And I don't want to say that to scare anyone or to suggest we need more regulation about them, but we do need to understand them and understand the mechanisms that drive those particular species to bloom. Then we have a couple of others. So Pluricentrum over here, it causes a lot of the mahogany tides that you might see if you were driving across the Dolphin Island Bridge, for instance. You'll often see blooms of that particular species. Sometimes, not always, it will produce toxins in the oxidaic acid class, and these are, again, producing diuretic shellfish poisoning. I'm not going to go into that, but I think you can probably predict the consequences of a diuretic shellfish toxin. It's not pretty, but not usually life-threatening either. There's been a huge impetus stemming all the way back into the 80s, and actually Leanne Flewelling from Florida Fish and Wildlife has a great, oh, she's in the back, okay, has a great slide that I've seen her present before, talking about really early records, even from the 1800s, of this being recognized. But some of the impetus in research funding that was developed was following a really unprecedented and really long bloom that happened in January 2005. This lasted all the way to 2006 off St. Petersburg. And this particular bloom affected Florida, Alabama, and the Mississippi coastline, and many of the fisheries resources associated in those areas. Lots of fish kills, manatee deaths, led to a really massive effort, I think, particularly across the Florida coast, to understand the drivers of these blooms and predictors. So a lot of work was stimulated after these events, and many others. So what are the factors that drive bloom initiation, progression? There's also factors that drive toxicity, movement, so physical processes that move these blooms, and then the factors that influence the collapse. Well, they can come under 4 categories, so I thought we could keep it a little bit simple. They can be biological, and an extension of that is physiological, meaning what the cells need specifically. Physical, so these can be salinity and temperature, physical forcing and movement within the environment, and then chemical drivers, which can include ocean chemistry, nutrients, and so on. What I've put here, and you can get hold of this, and I can share any references with you. This is from Cindy Heil, a paper from 2014, where she was showing this is a schematic showing a simple model of how nutrient sources and cycling can change from a no bloom sort of scenario to a small bloom and then to a larger bloom. The main thing I want to point out here is there's a lot going on. There are a lot of predictors, and I'm going to go through some of these. But it includes lots of different nutrient inputs from many different places. And that's really, really important to understand, important to monitor, and then important to plug back into our models, particularly when we have a freshwater dominated system here. So we'll go through that. one of the important drivers is something we call biological succession, but this is when we have a different bloom of something offshore, way out in the deep water, that looks often a lot like this, photo in the blue here. Looks like slicks on the surface, and it can be mistaken for Karenia many times by many people. But many researchers have shown that Karenia brevis blooms are often preempted by this bloom. And this is Trichodesmium, which is a cyanobacteria, very, very good at fixing nitrogen. So it can live and progress in nutrient-poor waters, but in fixing the nitrogen then provides this other nutrient source. You also have grazers, so other zooplankton that move in and start feeding on this bloom. And all of that produces a nutrient pool that can allow really nice conditions for something like Karenia to thrive if it's in the area. But the basic needs of Karenia, through loads and loads of research from lots of people much smarter than me, include, in general, like they like to be in a sea surface temperature of about 20 to 28. I'll admit, I'm not really a Fahrenheit person. I'm from the wrong end of the planet. But so I tried to put this in Fahrenheit for you guys, about 68 to 83 or so, and a salinity of about 20 to 35. And of course, they can survive in conditions above or below this, but they're not going to do well. So they're not necessarily going to thrive in those conditions. I thought this was important to point out because for those of you on the Northern Gulf Coast, you'll know we have a lot of low salinity events all the time. So salinity of 20 is often quite high for us. Depending on where you are, in the bay can be sometimes 5 parts per thousand in the morning and 20 parts per thousand in the afternoon. That can actually cycle hourly based on the data of our physical oceanographer at the Dauphin Island Sea Lab, Brian Zonkowski. We've seen these oscillations can be dramatic and very high frequency. So that can be a really challenging predictor in our environment. We also know that northerly winds and upwelling events bring nutrients to the surface. So these are offshore events that bring deep nutrients that are sort of closer to the bottom. They'll bring those to the surface, and then these north winds will push them towards the surface and to the shore, which will be needed for the Karenia to thrive. And movement to the coast is driven by current, and in our region, our current is driven by wind. So again, these are some predictors that we know and we can deal with. So what data is currently used in these other models? So there are several models, and some of them have been developed as local models. Some of the points that I've added to this slide were included in a model that was developed for Charlotte County in Florida. I think I've got that right. And that paper I can provide to any of you is this Kaki et al., the 2014 paper. But the variables most indicative of bloom timing and progression have been developed by a lot of scientists. So I put most of these things together and those that were relevant to our region. So the bathymetry is important. So the bottom of our bay and our coastal zone. What that looks like, the depth of the photic zone, so where we have light penetration and organisms living, is important. We have a fairly shallow system. So that means we have really good light conditions usually. However, we also have heavy river-dominated system, which I've mentioned a few times, which brings in a lot of sediment. All of that sediment and activity in the shallow system means that we distort the chlorophyll signal that can be detected. So chlorophyll through satellite imagery is detected as a fluorescence signal. And we have a lot going on. We can get reflectance from sediment in the water. We can get reflectance from freshwater inputs and so on. We have a lot coming from river systems that can fluoresce as well. So it can distort what's going on. This also happens off Louisiana. So I've seen several reports probably over the last decade where, and it's usually not the scientists, but it'll be someone in the media saying there's a huge bloom of Karenia in Lake Pontchartrain. And at the time, the salinity was about 2, and there was no way for there to actually be a bloom there. So my first question is, did you take a sample? Did you look? Because there's a lot going on in Lake Pontchartrain, but it wasn't a bloom of any kind at that time. So we just have to be cautious about how we use some of the satellite data. Some other things needed or important for models, of course, a wind direction, and you would have seen in the last presentation that's included in a lot of the plots and a lot of the slides. That's going to drive currents, which is driving the movement of these particular HABs. So that helps us understand where they are, how fast they might be coming here, and that can be predicted, but not always accurate because things can change, winds can shift at any given time. Sea surface temperature, of course, is important for Karenia and important for the models. Distance from the river mouth, this is particularly pointed out in Florida. Here, we would probably be thinking about discharge from Mobile Bay. We can also use river gauge information for other HABs that happen in Mobile Bay. Then ocean chlorophyll has been used a lot in these models. That's using the 3-band algorithm for MODIS. That information doesn't matter so much, but that has been a fairly good predictor in systems that don't have a lot of bias from sedimentation and plumes. So off the Florida shelf has been working really, really quite well for their predictions. Then the nutrients that are most critical for HABs, carbon, nitrogen, both organic and inorganic, and phosphorus, really important. So if we have, higher frequency, greater spatial scale sort of data of these nutrients, we might be able to get better predictions locally as well. And then more importantly, most of the models that are currently used for prediction, 1 day out, 2 day out, and 3 days out, combines all of the fantastic real-time monitoring data that's been collected by people across the Florida coast. We have some monitoring happening here, the best we can do, but we need to find a way with limited resources to expand that, increase the frequency, increase the spatial scale so that we can have more information, because we're not going to get to a predictive place monitoring once a month or once a week at just a few locations. So a great level of expansion is needed, but we also can't say that, well, that's the state's responsibility, because the state has limited resources and limited people. So we need to back them up, and we need to find a way to assist, augment, and help with getting that information if we want to have good models. Okay. So forecast the water quality monitoring data that has been put into these models. I won't go through much of this because I suspect Leanne could talk at much greater length of what's happening in Florida and how these models are used. But basically, data from samples physically collected will give an abundance of how many Karenia cells are in the water, and then shellfish testing happens to determine, the levels in those shellfish and if a harvest can be released or not. And all of that is combined with satellite imagery, and these conditions are monitored daily, usually, for what's happening locally, and then reported twice weekly. And in the form of these kinds of maps, I included this particular one because we see that there's actually a red spot up here in Northwest Florida, so we could actually be included in that data set. And then air quality is a bit different, and folks are going to focus on this today. It's not something I've been particularly involved in, at least in the last 15 years, but air quality monitoring and forecasts. There's a really great system in Florida in place where, again, daily monitoring of beach conditions are reported and recorded, and those reports are released twice weekly as well. That means that there's actually a site that anybody can go to determine, okay, I'm going to this beach, what do the conditions look like for tomorrow? And you can get a fairly good feel for what's happening. Those forecasts also take into account the water quality monitoring data that might be available to help them understand where blooms and other things are happening, and the satellite imagery showing severity, direction, and timing of those blooms. So accuracy of models. this is probably a point of contention, and it depends on your goal, right? And all of us here today probably have a different goal or a different hope or a different strategy or question. But local models, so one of the really solid scientific papers that has been reported on this was from Charlotte County in Florida. And this showed that the accuracy of those predictions on the same day were about 90%. So that's good, and that is a really good number, right? You would hope that you would get that kind of accuracy. And then for, if you move out 1 day, 2 day, and 3 day in advanced predictions, we have slightly less predictive capacity, so 65 to 70% accuracy. And this is actually really, really good, okay? But just let me say this. I know and I hear you, for those of you that are local and you have oyster farms and you're using or involved in any kind of fisheries resource locally, that's probably not enough to do something. You know, that's not much time to rally, to move, to do, you know, to really prevent an impact on your industry. So our hope is that together we can find a way, find out what is needed, and find a way to do better risk management so that we can get better predictive capacity and help you and help everybody try and predict these things a little earlier. I think that's possible because we've determined a few things about what we know locally, even though we don't yet have those models available. So we know this is actually similar in Florida, but we have dry conditions predispose the region for a Cape Brevis bloom due to increased salinity locally. This usually coincides with longer, warmer sea surface temperatures that are also conducive to those blooms. And this data, we have our great neighbours in Florida that have done such an awesome high-resolution job of predicting their blooms in those areas that we can look at winds that drive the currents and move those blooms to our coast. So all of those things are helpful. Locally, we also have significant alongshore currents and nearshore processes that dictate movement along the Alabama coast. We have a fairly small coast. And high rainfall events, which we get a lot of, can mitigate these blooms and actually make them crash. So this is my last slide, but I just wanted to point out to the needs for a local model really include the need for more data. And I know that you hear that from scientists all the time, but it's true, and we need your help to collect and get more data because we can't do that alone. You know, I have one lab at the Dauphin Island Sea Lab and I do monitoring, but I can't do monitoring across the entire coast of Alabama multiple times every day for every day of the year because we have a lot going on. But together we can probably do better than once a week or once a month. So let's think of a way that we can do that. So with stakeholder engagement, so with everyone that cares about this stuff, I think these conversations are really important. Then examining nutrient inputs and working with the community and managers locally to see if we can reduce nitrogen inputs into the local areas. This can help mitigate local bloom formation and blooms sustaining in the area. And then exploring other satellite tools and pigments with other colors, and some remote sensing people I hope are around that can discuss that a little bit more. Of course, people and funds to support all of these activities so that we can integrate into existing models and tools that have already been developed in Florida, maybe modify those and see what we can do to improve. With the goal being, at least for me, is a little more of this in a sustainable way so that we can have safe seafood. Okay.
How long does it cost in the environment? Once the bloom goes, so bloom,
Did everybody hear that question? How long do the toxins persist after the bloom? Okay.
So the toxins are quite stable. So unlike bacterial water things, you can't put them up. So that means they're pretty good at sticking around. But in general, for toxins that have accumulated in oysters, the oysters can depurate those within a couple of weeks, depending on the level. So if they got really toxic, it might take 4 to 6 weeks. If they were only a little bit toxic, they might only take, a week or two. In the water column, though, they will hang around in the dissolved form just in the water for a week or two, depending on, again, physical processes and water movements. We do know that the ferrotoxins combine to sediments that get taken up by macrophytes, so plants and other substrates, and they can stay around in that form for probably 6 weeks or so. That doesn't make it a seafood hazard necessarily, but it can hang around. In accumulation and depuration studies, usually there's full clearance even at maximum 10 times the safety level for consumption of these toxins in 8 to 10 minutes. So that would be the maximum level.
One other quick question.
Yeah, how long does it take for there to accumulate a level that might be harmful to humans?
That's another question that depends on the density of the cells in the water column. If you had cells that were at 5,000 cells per liter, which is sort of the trigger level locally, that would take 6 to 7 weeks, I think I calculated before, to get to a level that would be affecting human health. But the reason that level is important is that you can have a mass explosion of these cells. If that happens, you might get accumulation within 1 week or within a few days.
So for a 1 week accumulation, would you have to have 1 million cells per liter?
100,000 probably.
So really, it's 100,000. You have about 100. You have about a 1 week lag period for the risk of something.
If you were sampling daily, that might be helpful. But
Yeah,
if
you
you're
didn't
sampling,
see the 100,000.
right, because if you miss 100,000, you're putting people at risk, serious risk.
Thank you so much, Alison.